Hello guys, welcome back to Dr. Hill's Academy. Today we'll be discussing an aspect of glycogen metabolism known as glycogen synthesis. Stay tuned. What is glycogen? Glycogen is a polymer of glucose. So we have polymer of glucose and is a storage form of glucose. So in animals, glucose is stored as glycogen. However, in plants, glucose is stored as starch. Where is it stored? It's stored majorly in the liver. So we have in the liver and muscle. Although some other cells store glycogen, but majorly the liver and muscle are two tissues that store glycogen in large amounts. Now, the liver glycogen, so let's say this is the liver glycogen, is utilized for maintenance of blood glucose. So whenever you are fasting, the glycogen from the liver is broken down to give us glucose. However, the muscle glycogen is not usually utilized for blood glucose level. So the muscle glycogen, the muscle is very stingy. It utilizes its glycogen for energy within the muscle itself. All right? So the muscle glycogen is utilized for muscle contraction, while the liver glycogen is utilized for maintenance of blood glucose level. So this is what the structure of glycogen looks like. This is our glycogen over here. So you can see that the glycogen is a branched structure. So it is branched. Come on, it looks like a tree, right? Yeah, the glycogen is branched, looking like a tree, right? And then it should be compared with amylose, which is found in starch, that is not branched. So amylose found in starch is a straight unbranched polymer of glucose, unlike glycogen, which is highly branched. Now let's zoom in on this to see how the structure really is. So let's first see what a glucose molecule looks like. So we have the structure of a glucose molecule. And this is our carbon 1. These are carbon 2, 3, 4, 5. And then we have the last carbon, which is carbon 6 over here. Come on, remember, glucose is a hexose sugar, right? So there are 6 carbons. So this is the structure. Well, for those that like details, would put details for you. Remember, there should be OH here, OH, H, OH, then OH, H. For those of you that like details, right? Now we have carbon 6 over here. Don't forget. So I would use this structure to illustrate what we have over there. That's the polymer of glucose. We'll just illustrate the junction over there. Now we have the first glucose molecule over here. Always remember we have our carbon 6 here. So for simplicity's sake, I will not put the whole hydroxyl and hydrogen ions, right? So we have carbon 1, 2, 3, 4, 5, and 6, right? Then this glucose joins with the next glucose through a bond over here. I'll tell you what that bond is just in a minute. This is the second glucose molecule. And remember, we have our carbon 6. Come on. So if you notice, this is position 1, and then this is 1, 2, 3, 4, 5, 6. So this bond is between the position 1 of this glucose molecule and the position 4 of the next glucose molecule. What would be the name of this bond? It would be alpha 1 4 what? Glycosidic bond. Glycosidic bond. So we have alpha 1 4 glycosidic bond over here, right? Now the next one would be very similar. So we have a bond over here that joins with the next glucose molecule. So the next glucose molecule comes here, oh please bind with me, and then they bind together. We have our carbon 6 over here. So this is 1, 2, 3, 4, 5, 6. So you can see that this bond is also between carbon 1 and carbon 4. So it's also called what? Alpha 1 4 glycosidic bond. So these two bonds are alpha 1 4 glycosidic bonds. Now, between this molecule and the next glucose molecule that forms a branch, let's say we have another glucose molecule coming to bind. So we have, let me use red for the bond. So we have a glucose so this is carbon 1, 2, 3, 4, 5, and then don't forget we have our carbon 6. So this, for this glucose molecule that branched off from this linear one, it's binding carbon 6 and carbon 1. So this bond is called alpha 1 6 glycosidic bond. Come on, because it's between carbon 1 and carbon 6, right? This is alpha 1 6 glycosidic bond. Pretty easy, right? So the rest of the glucose molecules bind and the chain continues, right? So this glycogen is what we want to illustrate. So by the end of this lecture, you would be able to produce this glycogen, right? So glycogen synthesis occurs after eating, so in your fed state. After eating, you know, you don't want the blood glucose level to skyrocket, and then the liver mops up excess what? Glucose, and then uses this glucose to form glycogen, right? So the liver does a lot of work, you should think it. So after eating, we have glucose which has been digested and then absorbed into the bloodstream. This is our glucose over here. The liver takes up this glucose. So remember that it could be liver, it could be muscle, it could be any other cell, but majorly the muscle and the liver, right? So if this is the liver, which GLUT or glucose transporter should this be? GLUT2, come on. GLUT2 is usually found in the liver. So the liver uses GLUT2. Remember that the skeletal muscles use GLUT4, right? So you can check the video on glycolysis. I explained the glucose transporters over there. So the glucose moves from the blood into the interstitial fluid, and then from there it goes where? Through the GLUT, that's the glucose transporter, into the cell. Once it enters the cell, something can happen. This glucose, which has come here, so let's say this is our glucose molecule, glucose, can escape back. So it can move in and out freely, right? Depending on the concentration of glucose. Okay, so it moves along concentration gradient. Now, for the liver to trap this glucose, what does it do? it converts this glucose to something else known as glucose 6-phosphate, right? So what the liver does is to attach a phosphate to the glucose, right? So we have glucose 6-phosphates. That's a glucose with phosphate attached to what? Carbon 6, right? Of the glucose molecule. Now, which enzyme can do this? Glucokinase, right? If it's in the muscle, it is hexokinase. So remember that the glucokinase is found in the liver, hexokinase is found in the muscle, right? So we have our glucokinase slash hexokinase to form glucose 6-phosphate. This is our glucose 6-phosphate. This glucose 6-phosphate needs to be converted to something else. So we have the glucose 6-phosphate undergoing another reaction to be converted to glucose 1-phosphate. So we are moving the phosphate from position 6 to position 1, right? And remember that just as we have a kinase being any molecule or any enzyme that attaches a phosphate, right? A mutase is any enzyme that moves the phosphate from one carbon to another carbon. So we have carbon 6. We have a phosphate at carbon 6, right? So a mutase would take the phosphate at carbon 6 and move it to carbon 1. So that would be the job of a mutase. So we have a phosphoglucomutase, converting it to glucose 1-phosphate, right? Remember this reaction, this second reaction is reversible. However, the first one was irreversible. The first one required ATP, don't forget. First one required ATP and then gives off ADP. Remember, the second or the last phosphate is attached to form glucose 6-phosphate. And then this reaction 2 is reversible, and then it involves conversion of glucose 6-phosphate to glucose 1-phosphate. Pretty easy, right? Yeah. So moving forward, would illustrate glucose with something else. Remember that this is the structure of glucose, right? So we have carbon 1, 2, 3, 4, 5, and 6, right? So let's illustrate that with this bad boy over here. So this is a bad boy, and then see his arms over here. So this is his hand, these are his legs. So we illustrate, would represent this with carbon 1, and then carbon 4 and carbon 6. All right, so let me use the red so you can differentiate it. So let's say this would be carbon 1, that's the foot end is carbon 1, the head end is carbon 4, and then the hand end is carbon 6, right? You know, it looks like the structure of glucose, okay? Now, our glucose 1-phosphate will then look like this. The head with the legs, right? And then the hand, don't forget. So the phosphate is at the one end, so it's at the foot, right? The foot is the reducing end. The carbon 1 is the reducing end, and then the carbon 4 is the non-reducing end, okay? So the phosphate is at the reducing end, which is carbon 1. Now for this glucose 1-phosphate to start glycogen synthesis, what happens? It needs to be what? Energized. For it to be energized, it has to combine with an energy compound known as UTP. So let's say UTP. I'm sure a couple of you would have heard of this compound. Uridine triphosphate, right? When uracil binds with a ribose, it forms what? Uridine. Okay? So we have glucose 1-phosphate combining with a high energy compound known as UTP. So we have UTP over here. So when glucose 1-phosphate combines with UTP, what happens? The UTP gives off what? A pyrophosphate, right? So remember we have in total 4 phosphates, right? 1 here, and then we have 3 here, right? So 2-phosphate leaves as what? Pyrophosphate. And then we have what? 2 phosphates remaining, right? So the 2 phosphates would still be combined with the uracil, right? Or the uridine. So we would have a glucose here. This our glucose is smiling because it's entering the glycogenesis pathway. See the hands over here. And then at the foot end, we have what? UDP. So we have UDP binding at the foot end, right? So this is called what? UDP glucose. So we have UDP glucose. This is an energetic form of glucose, right? So it's in its energetic form. And then that's what will continue the glycogenesis. So this was the next reaction that was occurring, right? And then this pyrophosphate, what do you think happens to it? It's broken down to form what? 2 inorganic phosphates by the enzyme known as pyrophosphatase. So this pyrophosphatase does what? It separates the 2 phosphates bound together to form 2 inorganic phosphates, right? Yeah. So for the next reaction, we have this UDP glucose coming to combine with something. There's a bad boy over here. So what does this look like? This guy over here now looks like an anchor, right? So this guy looks like an anchor. He's called glycogen. It's an enzyme that binds with UDP glucose, right? So this enzyme, why it represented with anchor is that it serves as the anchor for glucose molecules to polymerize. So all the glucose molecules just bind to this anchor over here, and this is the what? Glycogen. So this step involves UDP glucose combining with what? The glycogen. So we would form our anchor over here. And then we have the glucose over here. So remember, this is the head and this is the foot end. So you can see that this UDP has left the reaction. So UDP is given off, right? And then in this glycogen is a protein, right? It's an enzyme. Enzymes are protein, right? And then within it, there is, as proteins are made up of amino acids, right? So there is an amino acid known as tyrosine. So there are various amino acids, one of which is tyrosine. So within this tyrosine over here, see the tyrosine residue over here. Within this tyrosine, the glucose attaches. So this glucose attaches to the tyrosine residue, right? The hydroxyl end of the tyrosine residue. That's where glucose attaches. And now, this step, let's say reaction 4, let's say this reaction 4, this step is known as autoglucosylation step, because the glycogen is auto-catalyzing this reaction. So it catalyzes the reaction on its own. It's a protein anyway, and then it has an enzymatic activity or ability, right? So it catalyzes the combination or the attachment of glucose to itself, okay? And then UDP, as uridine diphosphate, lives in the process. Okay, this uridine diphosphate, anyway, some of you would want to know what happens to it eventually. So the uridine diphosphate, UDP, can eventually go to combine, let's say UDP, okay, let's move it over here to avoid confusion. So the UDP can eventually go to combine with ATP to give UTP plus ADP. So you've seen that we formed UTP, which can now go back to energize more glucose molecules, right? Yeah. Now, this glycogen continues to act, allowing more glucose to bind. So the same thing occurs. That's the UDP glucose coming to attach to the glycogen. So the various glucose molecules come, attach, and then they just attach that way. So we would now form a molecule consisting of what? A few molecules of what? Glucose, right? So we have a glycogen with a few molecules of glucose, about 5, 6, thereabout. This is now called something known as primer. So this is what we call a primer. So in the next reaction, let's call this is the next reaction. Let's say reaction 5. What happens? In reaction 5, an enzyme comes to do the proper elongation. So the next step, that's step 5, is known as elongation. Because a bad boy comes and keeps on elongating the chain, this chain that was formed. You know, this was just a very short chain, right? So this was, the previous one was an alpha 1, 4. That's a short alpha 1, 4 glycosidic or glucosyl chain. Let's say glucosyl chain, right? That was the previous one. Now, this elongation involves what? The addition of so many glucose molecules. So we have our glucogen over here. So let's say these are glucogen, right? And then we have our glucose. So it goes on and on. So let's say various glucose what? So you can see that we now have a long chain of what? Glucose molecules, right? Attached to the glycogen. Now, the reaction was catalyzed by glycogen synthase, which is the enzyme glycogen synthase. It catalyzes this reaction, allowing for so many glucose molecules to be attached to the glycogen. So if you can see, we have a long chain, right? Then something happens here. What happens here? This is the next reaction, right? And now, this chain of glucose molecules are being cut, all right? So let's say these are glycogen, and then remember we had various glucose. Now, this glucose, remember that they are being held by alpha 1, 4 glycosidic bond, right? And then this end over here is the non-reducing end. This end is the reducing end. Remember I said carbon 4, which are represented with the head over here, is what? The non-reducing end. And then carbon 1, which is the tail end or the foot end, is the reducing end. So this is our non-reducing end. This is our reducing end. So this boy over here, there's a very bad boy that's, or should we say a lady? Yeah, a very bad girl. So she, on one hand, she has scissors. So on one hand, she has scissors, and then on the other hand, she has what? Gum or glue. So we have a cutter and a glue, right? So what she does is that she goes to the non-reducing end and cuts out about 10 glucose residues, right? Cuts out about 10 of them and then transfers them to another glucose molecule, but not to the terminal glucose molecule. Let me explain. So she cuts out here. Let's say this was 1 alpha 1,4 glycosidic bond. So she cuts the alpha 1,4 bond, and then she now transfers the glucose residue that was so gotten to another glucose molecule, but not to the terminal end, right? So we would have something like this. Let's say we have, so the bad girl would take that residue gotten and then attach it somewhere here. So we have something like this. Now, you can see that the bond we had here throughout this place, the bonds we have are alpha 1,4 glycosidic bond. But then this branching bond over here, so let's use the red to illustrate it. Over here, this branching bond is alpha 1,6 glycosidic bond, right? Just like I illustrated at the beginning. So we have the alpha 1,4 glycosidic bond and then alpha 1,6 glycosidic bond at the branching end, right? So the name of this enzyme is called what? Branching enzyme, right? That's pretty cool. So we thank God scientists didn't give it funny names, branching enzyme. But guess what? It has been given a funny name. So this is called 4,6 transferase, or you can say 4,6 glycosyl transferase, because it cuts the alpha 1,4 bond and then transfers it to alpha 1,6 bond. So it cuts an alpha 1,4 bond and then forms an alpha 1,6 bond. That's why it's called 4,6 transferase or 4,6 glycosyl transferase. So after that happened, what do you expect? The next reaction is a repetition of what? Elongation reaction. So that's glycogen synthase comes again. So we have glycogen synthase. So basically, the same thing occurs. That's the elongation occurs again, where the glycogen synthase would add more glucose molecules. So the glycogen synthase would come and start growing these molecules or this residue over here. So it keeps on adding, and then adding here, just like it was adding previously, right? In the elongation reaction. So elongation reaction occurs again to increase the length of these two. Okay. So let's assume this is the elongated form. So let's say this is the elongated form. And then this chain goes on and on to meet the glycogen, right? Then after the elongation, what happens again? The branching, all right? So branching enzyme comes, just like previously, it comes and then what does it do? It breaks off here and breaks off here, and then it transfers this short residue onto this non-terminal end and transfers this residue onto a non-terminal end as well, right? So what do we have? We have something like this. So you can see that the branching enzyme, that's our branching enzyme, that's your 4,6 glycosyl transferase, right? Branching enzyme cuts this, and then joins it to form a more branched form, which continues and continues until we have what? Our glycogen. And remember, the glycogen would still be bound to the glycogenine over here. All right. So as a brief recap, after eating, when glucose level in the blood is high, the glucose enters the liver cell or the muscle cell through glucose transporters, right? So the glucose is converted to glucose 6-phosphate and then to glucose 1-phosphate, which is now energized by combining with UTP. And then the UTP gives off pyrophosphate, which is PPI, and then forms the UDP glucose. And then the pyrophosphate can eventually be broken down to form two inorganic phosphates by pyrophosphatase. Then the UDP glucose now combines with glycogenine, which is your anchor over here. So the glycogenine now autoglycosylates the reaction, right? So it now forms a primer which involves glycogenine combined to a few number of glucose molecules, right? And then the next thing that happens is what? Elongation, where the glycogen synthase now starts adding so many glucose molecules, okay, to form a long chain. And then after which the branching enzyme now comes and then breaks off alpha 1,4 glycosidic bond, transfers the glucose residue so gotten, that's the glucosine residue so gotten, to a non-terminal glucose molecule to form a more branched molecule through the formation of what? Alpha 1,6 bond, right? And then the same reaction repeats itself, that's the elongation repeats itself, after which what? The branching occurs again. Thank you guys for sticking to the end. I hope you did enjoy it. Do well to subscribe, put your comments below, like and share to your friends so they can benefit from this lecture. Thank you and see you in the next lecture.